Good morning. I'm Gord Mills of the MD Anderson Cancer Center and co-director of the Khalifa Institute for Personalized Cancer Therapy. This morning I'm going to tell you about the program in personalized cancer therapy at the MD Anderson Cancer Center, both what we're doing, the challenges, the opportunities, and how we're going to be able to move forward in this critically important field. Now, this is my potential conflict of interests. My institution requests that I show this at all presentations. I think it's important to stop and talk about this for just a moment. First of these is that if we are going to help our patients, we need to develop a better way to work with industry, to develop academic-industrial collaborations that are functional and effective. So while we need to take care and ensure that these conflicts of interest or potential conflicts of interests do not become problems, it is critically important that we establish collaborations, that we move these forward, and that these become steps that go to helping our patients and moving drugs to our patients in a much more effective manner. And I'm going to talk about that at several points throughout my presentation. Now, the concept of personalized medicine or personalized cancer therapy is not new. Indeed, a fellow Canadian, Sir William Osler, stated as long ago as 1892, if it were not for the great variability among individuals, medicine might as well be a science and not an art. So the concept even then is that the variability among individuals in their own genome, and particularly in cancer and cancer genomes, had to be something that we dealt with in order to help our patients in the most efficient manner. Now, this is the mantra of personalized medicine, and it's one that has been around for over a decade. And I'm going to tell you that I've changed this or modified it slightly. The concept of personalized medicine has been to give the right dose of the right drug for the right indication for the right patient. But in terms of cancer care, the key term here is the first time. The first time we treat the patient is our best opportunity to get it right and improve the outcomes for our patients. Now, what I'm going to talk about today is really a revolution in how we think about cancer care. In the past, we have used relatively blunt instruments, chemotherapy, radiation therapy, primarily targeting the proliferative rate of the tumor. Now we have an opportunity to characterize each patient and their tumor with omics technologies, genomics, transcriptomics, proteomics at a breadth and depth never before possible. Indeed, we have entered the era where we can consider analyzing a complete genome, or at least the genome that is actionable in a cancer patient, for under $1,000. So the technology is here. The question is what are we going to do with it? And importantly, are we going to be able to use this technology to help our patients? Indeed, one of the opportunities of targeted therapies is understanding what is happening in the patient's tumor sufficiently to be able to capitalize on the vulnerabilities that that cancer has. It turns out that systems are robust. And indeed, I had a department of systems biology at the MD Anderson Cancer Center. And the concept behind that is that through phylogeny evolution, systems have become extremely robust to perturbations. Indeed, as a geneticist, it surprises me that any individual survives and grows to become an adult with all of the things that can go wrong. Further, the things that we do to ourselves, the toxins in the environment, the caffeine in the coffee that we drink, the cigarette smoke, are major challenges to individual cells. Indeed, the amount of caffeine that I've consumed already this morning, if put in a tissue culture dish, would kill each of the cells in that tissue culture dish. However, in the body, those cells have the ability to enter into a quiescent state or adapt to the stress that I have placed on the cell and wait until there's such a time as they can go back to function further. One of the opportunities in cancer is that they have lost those robust networks. The very underlying genomic instability that leads to the development of cancer may indeed be the cancer's Achilles heel. They no longer have robust systems that when we perturb them with targeted therapies, and in particular rational combinations of targeted therapies, that the cancer cell will have little opportunity or little choice but to die. Indeed, I think we're entering into an era that is incredibly exciting. We can now talk about the concept of letting the patient teach us what is important. Each patient has a story, and if we can just understand that story, we will be able to help patients like them. The concept of a patient like me from someone we've treated previously and had great results is moving forward at a rate that we've never seen before, and I'm going to come back on that. But there's another part to this perfect storm. The first that I've already mentioned is our ability to characterize the tumor in a breadth and depth never before possible with speed and cost efficiency. But the other part is we've now developed a drug toolkit. There are hundreds of drugs moving through the pipelines that target particular aberrations in patients' tumors. If we can just figure out how to use these optimally, how to develop rational combinations, and which patients we should treat with each of these combinations, we have an opportunity to move forward in a way that we've never had before. I want to point out that this targeted therapy toolkit is really quite impressive. There's almost 1,000 drugs either in or about to enter into clinical trials. Those drugs are spread across different diseases, but for example here, there's over 100 that are being evaluated in breast cancer and lung cancer alone. One of the big challenges is that we know that we need to do rational combination therapy, targeting more than one aberration in the tumor cell if we're going to have the success that we need. And if one simply thinks about the concept of testing 100 drugs and 100 different combinations, there simply are not enough patients to determine whether those combinations will work. And indeed, we have to move from the concept of a rational drug combination of two drugs from the same company to really where we start to look at two drugs that we put together because they take care of the challenges raised by each other. The idea that we treat the adaptation of the tumor cell to the responses. Now, I mentioned earlier this concept that we have to work with industry. What they do, what they develop, the drugs they develop are truly impressive. Indeed, there is approximately a $20 billion annual R&D investment in industry, in the pharmaceuticals and biotechnology industries. We need to capitalize on that effort if we're going to help our patients efficiently. Now, this slide is busy. I apologize just a little bit for it, but I'm going to show you some points in the slide that I think are very important. And the reason it's busy is that I built this slide many years ago when there were only 2 or 3 different targeted therapies that were in the clinic and were showing activity. But I want to show you a couple of messages that come from this particular slide. And the key one is that most effective targeted agents are linked to response prediction biomarkers. In the absence of a biomarker that says this patient is likely to benefit, or a biomarker that says that patient is not likely to benefit, we will have little activity and little utility. Now, what started this excitement was a drug called imatinib mesylate in chronic myelogenous leukemia that targets the BCR-ABL translocation. Unfortunately, or at least part of this story that is important, the BCR-ABL translocation was discovered in 1982. It took well over 30 years before we had an effective therapy approved targeting this aberration. However, the important part here is that imatinib mesylate and CML probably taught us a way, not the way we should do things, but away from what we should do. It was thought at the time that imatinib mesylate was introduced that if you had a disease, in this case chronic myelogenous leukemia, and a drug that matched that disease, imatinib mesylate, you would have incredible responses. What we forgot is that CML is primarily a disease defined by an underlying genomic aberration, this BCR-ABL translocation. Now, I want to skip down this list to a couple of different examples that I think are very important as we consider it. But I do want to just note that we have a number of highly effective drugs that are working across different diseases that have been approved, many not as quickly as we would like. But I want to drop down to crizotinib here, which came into use in lung cancer a little while ago. But what was exciting is that in lung cancer, a group in Japan identified an aberration of a fusion gene called EML4-ALK. ALK happens to be a kinase, very much like the ABL aberration in the BCR-ABL translocation. So it was thought if we could find a drug that would target ALK, we would be able to move this forward to the clinic. And really what was very exciting is that crizotinib, a drug that was being developed for a completely unrelated reason, and indeed using an antibody that I developed many years ago, was shown to be active against ALK. This led to a large clinical trial, and indeed from 4 years from the time of discovery of the EML4-ALK fusion gene to implementation and approval by the FDA. So this is a massive switch from the 30 years from identification to therapy in the imatinib mesylate story to that of crizotinib, 4 years in the current era. It is hoped that with this drug toolkit and the ability to match patients, we will see many more examples of this rapid translation to the clinic and ability to help patients that we have not seen previously. Now, I want to note that almost all of the drugs on this list target what we call oncogene addiction. The tumor has an underlying genomic aberration that drives the behavior of the tumor. They are addicted to that driving event. And indeed, as long as we can identify an addicting event, we have a targeted therapy with an adequate therapeutic index, we can expect to have marked activity in patients' tumors. There are 3 exceptions on this list. One that I do want to bring up are the PARP inhibitors. BRCA1/2 are major players in DNA repair. There are a limited number of DNA repair pathways, and a patient with an abnormality in BRCA1 or BRCA2 has defective DNA repair with limited options left to repair the damage that happens every time a cell divides. If we instead target one of the other pathways for DNA repair, one that is mediated by PARP, we can now induce a synthetic lethality. BRCA1/2 mutations do not harm the tumors. In theory, PARP inhibitors will not have major side effects on dividing cells, but when you put those two together, only the tumor cells have the abnormality, and you will obtain, hopefully, a synthetic lethality that results in a benefit to the patient. And indeed, in a number of trials, including ones that we are running, we have seen remarkable activity, particularly when we use these PARP inhibitors in combination with other drugs. But I do want to go down to 2 of these at the bottom very briefly. And that is that the most effective targeted therapy, and indeed the one that we have used for the longest time, is an androgen receptor inhibitor, tamoxifen and AIs in breast cancer, targeting a lineage event, an expression of the estrogen receptor that is important for the lineage of cells that become luminal breast cancer. So we're not targeting something that is related to the tumorigenic process, rather a critical event in the differentiation of the cell of interest. And indeed, chronic lymphocytic leukemia, targeting BTK, a key regulator of the B-cell receptor signaling, gives you the same types of responses, and these are targeting lineage events. Now let me point out that there are a number of other targeted therapies where we don't have effective biomarkers, PAGFR inhibitors, proteasome inhibitors, and indeed I'll contend the limited activity of these drugs is related to a great degree from not being able to identify patients likely to benefit. And I'm going to come back to this concept that I've pointed out here, that the efficacy of a drug requires a predictive biomarker, and that those predictive biomarkers are a combination of the underlying driving aberrations in the tumor, the aberrations that occur in concert, the co-mutations, and the lineage of the tumor cell. Now, this sounds great. However, there are challenges, and I'm going to talk about the challenges at a number of different levels. The first is that their response is in only a subset of patients. Our biomarkers today have strong negative predictive value. That is, less than 5% of patients without the biomarker can be expected to respond. In contrast, the positive predictive value is much weaker. Somewhere between 30 to 60% of patients with the biomarker are expected to respond to these drugs. And the biggest problem is that most of these responses tend to be short. There are many different resistance mechanisms, and if we're going to have effective targeted therapy, we're going to need to figure out how to target those resistance mechanisms. Unfortunately, resistance appears to be dominant over sensitivity. One of the best examples of this is the epidermal growth factor receptor and RAS mutations. If you have an epidermal growth factor receptor mutation, you will be sensitive in most cases to drugs targeting the EGFR. However, if you have a concurrent RAS mutation, and that's not an unusual event in bowel cancer and even lung cancer, you will have limited benefit from the drug. Similar examples have come up with HER2-targeted therapy, with aberrations in PI3 kinase and PTEN, as we showed many years ago, and more recent data showing that if you have a co-aberration in the MET oncogene in HER2, you are unlikely to have significant benefit. Something that I'm not going to dwell on to a major degree today is the other aspect. I mentioned earlier that a therapeutic index is critical to what will happen in terms of being able to target a molecule of interest. It is the patient's own genomic makeup, not the makeup of the tumor, that determines the metabolism of the drug and the toxicity to the individual. And that defines the therapeutic index in concert with what is going on in terms of the tumor responsiveness. I said I'm going to talk about the challenges in many cases. And indeed, I think that the challenges are greater than we initially thought as we entered this field. And a real question, I think, has arisen as to whether we can truly achieve personalized cancer therapy. We run into an N of one problem. Every tumor is different. Every patient is different. Can we obtain enough information and enough commonality so that we will know which patients are likely to benefit from each drug? Indeed, I think we're moving to an era where we talk about precision medicine or stratified medicine, treating more and more homogeneous patient groups. Unfortunately, that means smaller and smaller groups of patients are likely to benefit from any particular therapy. As we move forward, this can be a trouble or a problem in proving that the therapy that we're interested in will actually benefit our patients. For example, ductal breast cancer, the most common cancer in women, has now been demonstrated to be a minimum of 8 independent therapeutically relevant subclasses. Instead of being one of the easiest diseases to perform clinical trials, it is now a set of orphan diseases. And indeed, in one of the aberrations or one of the subtypes of breast cancer that we identified the aberrations for, Claudin-Low or metaplastic cancers, there are particular aberrations that could be targeted therapeutically. However, it has been very hard to mount a trial to prove that those targeted therapeutics work. The other challenge that we are entering into is that the aberrations that we want to target are particularly rare across tumors, or may be sufficiently rare as to make this challenging. And I'm going to talk about this in several examples as we move forward. One of the molecules that I work on, called AKT, part of the PI3 kinase signaling pathway that I'll show you in a few minutes, is mutated in perhaps 2 to 3% of breast cancers. However, as we look across patient sets in our clinics, we find that under 1% or approximately 1% have an abnormality in AKT. Now, if we had a drug that worked in 1% of all cancer patients, that would be wonderful. The challenge, however, is how do we determine or identify those individuals? The other that we are finding as we move forward and have testing opportunities that we can look at many different things in a tumor is that the amount of tissue that we have frequently is limiting. That the limited amount of tumor can result in challenges to test all of the different events that we need to do. And as I mentioned a moment ago, resistance is dominant. One of the ways in which we are dealing with this problem and trying to understand how to move forward is to doing prospective analysis of many different genes at the same time, amortizing the costs of testing all of those genes at once across a large number of patients to determine what patients are best to go in which particular trial and which drugs may benefit our patients. Now, we've been doing this in the Khalifa Institute for Personalized Cancer Therapy at the MD Anderson Cancer Institute. And we have a mission and vision that I think is important, and I'll start with the mission, and that is that we're going to determine how to personalize cancer therapy for all of our patients. I'll mention how much of a challenge that is in a moment. But one of the key things that we need to do is to define the new standard of patient care. And that is only going to happen if we show that we attain durable responses leading to cures, or at least the equivalent of cure, by converting cancer into chronic disease and reducing the costs of patient management. These processes are relatively expensive when one looks at this in total. However, when we look at each individual patient or each individual gene that we are testing, the costs really are modest compared to what it would be if we were looking at doing a CAT scan or any other similar analysis of the patient. But if we don't determine how to make this billable and payable, we're not going to move it forward. Now, at the MD Anderson Cancer Center and the Institute, we see approximately 30,000 new patients a year. This is an engineering problem to characterize what is happening in their genome broadly. And indeed, we're looking probably at testing and analyzing at this point in time somewhere between 4,000 and 6,000 patients a year. What we want to do is to characterize what we call actionable aberrations. And I use the term mutations here to refer to anything that happens in the genome. Actionable is something that we can do something about. One of those, of course, would be if we had a targetable event, an aberration that we could target either directly or by targeting the pathway that it influences. We would also want to know whether they would predict patient outcomes. Could we have a prognostic indicator that would allow us to improve patient management? The technology has to be paraffin compliant because that is the tissue that we have available to us, and I'll talk a little bit about the need for proximal biopsies as compared to biopsies that we did many years previously. For those patients where a targetable aberration is present, and I'll show you some numbers in a few minutes, many of them will have standard of care drugs, drugs targeting the EGF receptor in lung cancer, BRAF, and melanoma. What we use this for most efficiently is to fill clinical trial cohorts, to test the concept that if one has a PI3 kinase aberration, will a PI3 kinase targeted drug work or not? We can answer those questions quickly, and where the answer is yes, we get those drugs through to our patients in a much more efficient manner and actually let everyone in the world benefit from this information. The other that we do, which is much more challenging, is test the process. Ask whether going through the process of obtaining this data, matching the patient to a drug based on the underlying abnormalities in its tumor, will benefit the patient. This is not to ask whether each drug works, not to ask whether there is a link of one drug to one particular aberration, but rather to test the process. And this has become a challenge and an opportunity. Unfortunately, for many of our patients, when we look at an actionable genome, we do not find an aberration that we can act upon. And in many of those cases, we will characterize those tumors in exquisite depth as a learning process to let the patient teach us what is important. One of the key components of that is not only do we identify the aberration, but we determine whether it is a driver by high throughput biological validation, a driver being something that drives the behavior of the tumor. And if we had a drug for that driver, we could benefit the patient. As we attain more and more information of that type, we can go to the pharmaceutical industry and hopefully convince them that this is something they should build drugs to in concert with academic efforts. Today I'm going to use the PI3 kinase pathway as one of my major examples as we move forward to show you some of the principles. The PI3 kinase pathway links the membrane through a number of signaling events. This is PI3 kinase itself. This is the AKT I mentioned a few moments ago, PTEN, the major negative regulator, all the way through to almost all of the important things that are happening in the cell. This particular pathway is aberrant in more patients, in more diseases, in terms of activation than any other pathway. Targeting an activating event is relatively easy, and indeed this pathway is chock full of kinases that we know how to target efficiently. This has not escaped industry. These are just some of the drugs that are currently in clinical evaluation from different companies asking what is the best drug in terms of pharmacology, what are the best targets in the particular pathway, is it PI3 kinase itself, is it AKT, is it mTOR, where would you like to block the pathway, at the top with a growth factor receptor inhibitor, or down at the infector arm, say at an mTOR or a 6 kinase inhibitor. These questions are being asked rapidly and efficiently, and we should have answers for many of these soon. I want to show you some of our early data from Janku et al. It's already published from our phase 1 program that gives us, I think, some important lessons to pay attention to. So this is a waterfall plot, patients that benefited over here at the right, and the patients that really their tumor kept on growing at the left. And what we have is these diseases are coded by the lineage in color. And I want to note that all of these patients were tested with multiplex testing, as I mentioned previously, and all of them have aberrations in a single molecule, PIK3CA. Now, in a phase 1 trial, this is remarkable. With a RECIST criteria of 30%, we have over 40% of our patients benefiting. So linking a drug targeting the aberration seems to be highly active, as we would expect less than 10% to respond in any normal phase 1 trial. However, I want to point out, I think that there are much more important lessons in this particular figure. The first is that these patients over here on the left are red. They're bowel cancers of various types, and in reality, they do not seem to benefit from these drugs despite having the PIK3CA mutations. Now, I mentioned earlier that bowel cancers have frequent aberrations in KRAS, and we thought that would be the problem. And indeed, some of these patients do have abnormalities, co-mutations in KRAS, but that doesn't seem to be the sole reason why they're resistant, because over here we have patients with mutations in KRAS and BRAF in ovarian cancer that benefited from the PI3 kinase aberrations. Indeed, as we look at this group, it happens to be the set of women's cancers that I work on: cervix cancer, endometrial cancer, breast cancer, ovarian cancer, and we're attempting to understand why these tumor lineages are particularly sensitive to these targeted therapeutics. I just want to show you a couple of pictures to show that this is real. Here is an endometrial cancer in a patient who had had extensive treatment previously, and after 18 weeks, this has had a massive shrinkage in size. Here's an ovarian cancer with 2 pelvic masses, one here, and after 18 weeks, this one is decreased markedly, and this one is essentially scar tissue. So this really does work even in phase 1 trials with single agents, suggesting that if we can just figure out how to do this better and what rational combinations to use, we have the opportunity to benefit our patients. So with this in mind, we have done a major effort in our acting program that we call Clearinghouse, wherein patients are identified by the physician, and I apologize for not updating this. This is a little older slide. We have now tested over 6,000 patients in the program. We obtain tissue, usually from a previously existing paraffin block, a biopsy if it's clinically indicated. We then do CLIA testing in our pathology and lab medicine environment using a hotspot analysis of either 46 or 50 genes now, followed by, when it's appropriate, full-length sequencing of 400 genes that have been implicated in cancer. As I mentioned earlier, we're looking for actionable events. We have a database development that allows us to link these aberrations to patient trials that is critically important. We have a queryable database of patients and mutations and a decision support tumor board. And I'm going to come back to how important it is to provide information to our physicians that links this incredible trove of new information through to how you would help manage patients. And we do an enormous amount of data capture, not only to ask whether this works, but how would we do this better in the future. And I'm going to talk a little bit about the number of patients that we would expect to go on trials in one of these programs. We do have major research programs going on in parallel with this, where we more extensively characterize what is happening in the patient's tumor in the non-CLIA environment, again, to design our next trial. Now, I want to show you some results from the first 2,000 patients that we analyzed at the end of last year. This is based on hotspot mutational analysis, and what we find is that about 50% of our patients have a potentially actionable event. Now, I want to note that we do not count p53 as actionable, although there are some particular drugs and mutations that you can test. It's not generally something we can do anything about. It's a tumor suppressor, and targeting lost events is hard. We have counted KRAS, and I think this is somewhat controversial. KRAS may or may not be targetable depending on what you're looking at, but we do have studies today that are looking at targeting KRAS in a number of different diseases. And there's a major effort at the NCI today to develop drugs that will target KRAS. But if we look at these mutations, you'll see that almost half of our patients have nothing that we can target with drugs available today. p53 is the most common. And then we come into a suite of targets: KRAS, PIK3CA, BRAF, IDH1, NRAS, for which drugs are available, and these are in the 5% range. But I want to point out that we have a tail of things that are important but potentially problematic. This includes, for example, the AKT that I talked about earlier, PTEN, EGFR. In the whole population that we're looking at, these are sufficiently rare that it is going to be difficult to prove that we have drugs that work. Now, in some tumor lineages, these are much more common, making the processes easier. But I want to show you the example of AKT once again. So AKT is mutated in about 1% of cancers overall, 2% of breast cancers. So we decided we would go to a company that has an AKT drug. There are actually 4 different companies that have AKT drugs together, and offer to do testing of patients and enroll patients with aberrations in AKT on the clinical trial. So for example, if we tested 2,500 patients, we would expect to enroll about 25 patients. This is not the number that you would predict from the percentage of AKT, but rather from the fact that many of the patients would not be trial eligible, some would reject the trial. And it turns out that this would be about $800 per test in our laboratory, and it would be $2 million just to do the testing. Now, the company looked at this and said, that's wonderful, you've got to be kidding, not going to happen. However, as I mentioned, we already do multiplex testing of all of our patients in this pathway, and we've in this process, and we've actually identified 29 patients with AKT mutations across different diseases, most of them breast cancer, as I mentioned earlier, and those patients are now being enrolled in clinical trials specifically designed to ask, if we target AKT, will we benefit our patients? Now, I also want to note that it depends on what tumor or what disease you are looking at. And so once again, if I take that same concept of what patients have potentially actionable events, when we look at melanoma and skin cancer, it's almost 75%, colon it's 70%, and we get down to esophageal cancers, sarcomas, prostate cancer, it's really a much lower event. So all of these things that I'm mentioning mean that we need to start thinking about targeted therapy as a combination of mutation, lineage, and the co-mutations that generate resistance. And looking at any one of those in isolation is going to make our life much more challenging. Now, one of the things that we're developing from this information is a series of umbrella trials. Umbrella trials are where you test all of the patients with a particular disease and then work with industry, this academic industrial collaboration that I have been talking about, to put in place a comprehensive drug approach to all of the aberrations that you have in the tumor. And the idea here is that you amortize the cost of the testing, which is non-trivial, over many different trials. Many of you may have seen these types of figures for lung cancer, and this is where it is best developed. But I do want to point out that if you were to do this in breast cancer, our example and one of the umbrellas we're building at the MD Anderson Cancer Center, over half of the patients have a targeted therapy, quite a broad list that one might use. Now note, about 30% of breast cancer patients have no actionable aberration. About 30% of them have p53 as their only targetable aberration, And we have the big ones, PIK3CA, other parts of that pathway, but we do have rare events and many other aberrations where we have drugs, BRAF, KRAS, things that drugs are in the clinic or available that could be used to test the hypothesis. Now, these particular trials, I think, are critically important, but come with a number of challenges. First one is how do we cover the cost of testing thousands of patients? How do we get all of the drugs from industry, getting different companies to collaborate with these drugs? How do we obtain proximal biopsies, biopsies of the disease we want to treat? And finally, what are we going to offer patients without actionable mutations? If we have to tell half of our patients that this isn't going to benefit them, will they enter on the trials? We have another type of trial, which is called a basket trial. And a basket trial is a concept where you look across many different diseases for a single aberration and then test whether that will work. And I already mentioned that concept around AKT1, but I want to show you one more and just mention these others very briefly. And the first one is BRCA1 and 2 that I mentioned. We have PARP inhibitors that look highly effective. BRCA1 and 2 is primarily found to be aberrant in breast and ovarian cancer, and we've removed all of the breast and ovarian cancers from this analysis. But as we look at the patients that we see at the MD Anderson Cancer Center, almost 10% of them have a definable aberration in BRCA1 and 2. No one disease has enough to make a practical trial. However, if we combine this, 10% of all cancers, it is very worthwhile if we can identify patients who will benefit. ALK mutations, 30%, AKT, 3%, AKT, the 1 to 2% we talked about, TSC1 or 2 in the PI3 kinase pathway, mTOR, and AKT amplification, all of these in the PI3 kinase pathway add up to a significant number of patients across many different diseases, but best done as a basket trial, not asking which drug and which disease, but rather, does this drug, when linked to a particular target, show efficacy? Now, I think there's an important concept and challenge that we need to list. I've showed you that many of the aberrations that we see in patients do not have drugs that are effective today. So if we were to go in and just ask a simple question of how many patients could possibly go on trials and what actually happens at the MD Anderson Cancer Center, we find that if we were to do hotspot mutational analysis of the 50 genes most highly implicated in cancer, the theoretical number of aberrations, and I'm taking out both P53 and RAS here, is 35%. Of those patients, at any one time, we only have trials at the MD Anderson Cancer Center for about 20%. And finally, if we ask what number could even theoretically go on trial if we put these two together, it's about 11%. So you will test 100 patients to be able to put 10 patients or 11 patients on a clinical trial linking their aberration with what is going on. Now, given that only 4 to 5% of patients go on trials today, this is a remarkable improvement, but it really leaves us wanting to do better. If instead we characterize a larger panel, 200 genes or so, and sequence the full length of those complete genes, anything that is actionable, rather than just looking at hotspots, our theoretical number jumps to about 53%. For the total population, about 30% would have trials, and an actual number where we would expect about 17% to go on trials. That is a great number. Now, I want to note that the rate-limiting factor in this is having the targeted therapy trials. And despite the fact that we have 900 trials open at the MD Anderson Cancer Center today, we're looking at really less than half of our patients with an aberration having a trial for that particular aberration. One of the other challenges that we will have to deal with as we move forward is when we test patients, we will not only find what is happening in their tumor, but also what is happening in their germline. We will find hereditary abnormalities in a number of genes, such as BRCA1, BRCA2, P53, P10, that are high penetrance, autosomal dominant mutations that have very major implications for the patient and their family. And each institution that does this is going to need to develop an approach to convey this information to patients. We really have limited recommendations for how to do this today, but it is something that needs to be on our horizon. We will also see things in pharmacogenomics. I mentioned earlier what dose of what drug to use. And finally, what do we do when there are other problems that are related to the aberrations that are not cancer? For example, hypercoagulation states, unpreventable diseases, for example, a predisposition to Alzheimer's disease, something that we can do nothing about. Now, one of the things that I've pointed out is we are generating an incredible amount of information on every patient. And this information is something that we weren't taught how to deal with in medical school and is evolving at an incredible rate. How is it that we are going to develop medical decision support that is going to tell us what to do? The past, our patient has expected the physician to know everything. How are we going to take this barcode of information, the incredible trove that we can get from omics technology, and tell us what to do for that patient? It is not as simple as laying back and being read on a barcode scanner. At the MD Anderson Cancer Center, we have developed a personalized cancer therapy website that will help physicians in this process. You can find this at pct.mdanderson.org. It is publicly available. You can also search on personalized cancer therapy with no spaces between those words on Google, and it'll take you to the site. This gives us a lot of information. This site will continue to be developed and matured. Right now, we have information on the 14 most important potentially actionable genes. You can look for what happens with mutations, copy number changes, fusions, germline alterations if relevant. It is interactive. The physician can determine how much information they want to attain from a very superficial view of saying, what clinical trial could I use for this patient, to going all the way down to the in-depth analysis of each paper, saying why we gave you the conclusions that we gave you. And the key is that we're telling you the therapeutic implications, not of an aberration in the gene, but of the specific abnormality that your patient has and the level of evidence that we have to tell us what you should do with this information. It is also linked, at least in the United States, to clinical trials that are available, not only at the MD Anderson Cancer Center, but anywhere else in the country that we're able to track and find. I want to tell you a couple of stories, and I apologize, a couple of my slides have little glitches in the Mac IBM switch here. This is a fairly old story, but around the concept that the patient can teach us things that are very important, and we finally have the tools to learn from those patients. I mentioned earlier that 5 to 10% of patients in early clinical trials demonstrate remarkable responses. And we started this particular study a long time ago, just as BRAF was being shown to be an abnormality of importance in melanoma. There was a drug available called sorafenib that was thought to be a good drug targeting BRAF. It turns out it's not. It's not a particularly good BRAF inhibitor. And we did a trial of 22 patients. One of those patients responded remarkably with their disease going away and staying away. And so the question is what can this patient teach us? Is this patient teaching us that they had an abnormality in BRAF because it was a melanoma, and that's why they responded? Or is there another lesson? And indeed, we went in and characterized this patient in great detail. There was no abnormality in BRAF at all. This is the results in this patient. I do want to show you that this really is remarkable. Here is a metastatic lesion to the lung. This is the lesion after 2 months of therapy. Importantly, 8 years later, this lesion is still gone, and the patient is essentially cured of their melanoma. That is remarkable. It's surprising. So we needed to learn what this patient could teach us. So we did the in-depth characterization that we talked about, and there was no mutation in BRAF or any BRAF family members. We went back to the drawing board and said, what's going on? Well, we knew that sorafenib targeted kinases, so we looked at all of the possible kinases and found what was clearly a causal mutation in KIT. This was a mutation that is rare in other diseases that have abnormalities in KIT, L576P, doesn't matter what it is, I'll mention it again in a moment. But following this initial patient response, we went in and characterized melanoma extensively and found that 30% of acral mucosa and chronic skin damage melanomas have abnormalities in KIT. And this is now part of our standard approach for these patients. And further, they receive KIT-targeted therapy, and I'll talk about its efficacy in a few moments. What made this even more exciting is this patient came in about 2 weeks after we had completed this study and found that KIT was important and that we had a drug targeting KIT. Turns out that dasatinib is a little better drug than sorafenib for this approach, but here's the patient, and this is a PET scan of their pelvis. This patient was such that we had to get special permission to treat them because their performance status was minimal. This is after 1 month. This disease is quiescent. The patient's improvement in quality of life was remarkable. We did not cure this patient. However, it was a beautiful proof of concept that this process was important and that indeed we should expand it further. And we have a major program at the MD Anderson Cancer Center, and we will work with anyone in the world who has similar patients. Any patient with a marked response in clinical trials. Here we're talking about complete or near complete or a mixed response. One lesion goes away and another stays. We will obtain a biopsy of the patient's tumor at response and at recurrence and then perform an extensive analysis. Initially, we'll look at those candidate genes and ask if the expected biomarker is present. Wonderful clinical validation of our hypothesis. But if not, we will move on to deep sequencing and determine what is happening. We believe that this is going to be the best way that we have to identify new targets and new drugs and biomarkers that will link the target to the drug. I want to go through a couple of other challenges fairly quickly. And the first one of those is, what do we do when we are treating a patient? Unfortunately, today we treat patients based on the original biopsy. What we're worried about is the metastatic tumor because we can usually remove that local tumor. And does that primary tumor represent the metastasis? And indeed, in many cases, we are treating a recurrent tumor that is not only spatially, but also temporally distant from when we obtained the original biopsy. So does the primary tumor represent the metastatic or recurrent tumor? Are there subclones? Are they selected? And does it matter? This is a beautiful paper from another group published in the New England Journal on intratumoral heterogeneity in renal cancer. And what I just want to point out is if you look at heterogeneity within the original tumor, in different sites it is present, and if we look at different metastases, we find that there are mutations that are quite different in each of these sites. However, when we look at that in a little more detail and ask about evolution of the tumor, we find this. And we now have a study from the MD Anderson Cancer Center with many more patients showing that this concept is right. Going from normal tissue to cancer, there is a truncal event, a major dominant or dominant series of events that, if targetable, would affect all of the tumor cells. The other that is important is that although you have branches, many of these branches have convergent evolution. This particular tumor has VHL as its truncal event. But note, in all of the branches, there's an abnormality in another gene called SETD2. So that SETD2 abnormality is important and co-occurs with VHL in every tumor cell that this patient has. However, once you get out to the mutations that are happening distally in the leaves, you may only affect part of the tumor. For example, here is an aberration in mTOR that is only found in some tumor cells. Here's one in KDMSC. These are different mutations, and it turns out that over 90% of the tumors will have at least 3 common events. So if we can identify these dominant events and target them, we would be much more efficient. We've looked across a large series of breast cancers with a number of questions, and first of them is that at least 80% of the breast cancers do not appear to have major intratumoral heterogeneity in targetable events. I'm not saying they don't have heterogeneity, but rather those things which we target tend to be truncal events and dominant, and so we could move forward with them. Now, one of the questions is, does it matter? And so this is an early analysis that we did just asking about the prognostic impact of a very simple question, ER, PR, HER2, and ask whether there was a discordance between the primary and the mets, would it matter? And this is if you have the same result in the primary and the met, and if you have discordance, it is a much worse tumor. So the very fact that you have heterogeneity indicates a worsened outcome. This is another study from the MD Anderson Cancer Center asking exactly the same question with HER2, and once again, exactly the same conclusions. Concordant, you do better than if it is discordant. So having multiple clones in the tumor is an indication of a worsened outcome in and of itself. This particular study indicates that it's not treatment, it is actually prognosis. This is a very interesting case from colleagues in Denmark, where they had a breast cancer, similar to ones we're looking at, where they looked only at PI3 kinase, and they found about a 50% discordance between primary and metastases, similar to what we're seeing. But they had an odd observation, where the primary had no mutation in PI3 kinase, the metastasis had this mutation, second metastasis had this one. And what they found is if you put a needle in, or were to put a needle in, this area in the primary tumor was wild type. This had one mutation. This had the other mutation. This is a case of convergent mutation. We have a paper from MD Anderson, not my laboratory, in Press, that suggests if you just very deeply sequence any place in this tumor, you would get all of this information. The depth of sequencing then matters. I also want to point out that we have one other opportunity that may help us, and that is looking at circulating DNA. Tumor cells release DNA from the tumor into the circulation. We can measure that, we can determine abnormalities, and we may actually be able to go in and, by looking at what is in the circulation, capture information on all tumor types in the patient. Our studies right now show somewhere between an 85 and a 90% concordance between what we find in the primary tumor and what we find in the circulation, suggesting that this may resolve many of the problems. So we are going to have to deal with intratumoral heterogeneity. Biopsy, biopsy, biopsy is a critical point. We're going to need do this with circulating DNA and circulating tumor cells. Now I'm going to finish off very briefly with just a couple of comments, and I'm not going to walk you through the slides. But one of the challenges that we have when we look at data from a patient is determining whether the aberration that they have as a driver, that is, if we were to target it, would behave or change the behavior of the tumor, or is it a passenger? Having that information is critically important because one does not want to target passengers. It will not be effective. Boris Bastian did a clinical trial to ask this question, and indeed, where he found that particular mutations, again, this KIT mutation that I was talking about earlier, that 576P, is a driver. 40% of the patients will respond. If you have another mutation in KIT, but it is not a driver, it's 0. So we need to know not just what the aberration is in a gene, but whether the aberration is a driver or passenger. The future is coming. It's going to be biopsy, 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 imaging, blood, circulating tumor cells to determine what's happening in the patient. Characterize these to the depth that we need to identify subclones, perform a trial of therapy, biopsy recurrences, determine why they recurred, and select your next therapy. Hopefully this will result in marked benefit for our patients. Thank you. Are there questions? Okay. So there are a number of questions. I'm going to go through as many of these as we can until the termination of the time that's available. However, if you have further questions, you're welcome to send them to me by email, and I will answer as many as I can. It's gmills@mdanderson.org. The first one of these is, are there any new developments with GI, colon, CML? I think one of the most exciting developments now is that we understand why BRAF inhibitors are not effective in colon cancers with mutations in BRAF, and we have combinations of drugs that are moving forward that will help this particular process. Now, the second question, I think, is one that I alluded to earlier around the concept of how the patient's genome determines drug metabolism and therapeutic index. Should we be analyzing the patient's genome to determine what drug to use and also what dosage of drugs? And while I would say that this is probably one of the most critically important things we should be doing, so far this has not been effective for most of the drugs that we use. And I think that this area of pharmacogenomics is one that is going to require major effort over the next 5 or 10 years. Now, I have another question here, which is around the concept of personalized stem cell therapy, and I'm going to add in the combination of personalized immunotherapy over personalized drug cancer therapy. And where do I think we are in terms of those approaches? And one of the things I do want to mention is that it is incredibly exciting that the promise of immunotherapy, which has been around for generations, is finally beginning to be realized through checkpoint therapy. However, even there, it's only a portion of patients that benefit. My expectation is that as we move forward, we are not going to stop doing chemotherapy and radiation therapy. They're extremely effective for a population of patients. We'll do a better job of knowing who is going to benefit. We will then look at the best way for each patient to use the armamentarium of targeted therapies, stem cell therapy, targeted immunotherapy in each individual, and I'm going to predict that the combinations of targeted therapies and immunotherapy are going to be extremely effective, more effective than either one of these alone. The next question is one that I alluded to briefly, which is the concept of how do you determine whether a mutation is a driver or a passenger? That's another full talk that I give in other environments. One of the obvious things that one can do is ask whether this aberration is commonly seen and has been characterized by others to be a driver event in cancer. And that's probably the best way that we have today. V600E mutation in BRAF, mutations within the region of V600E are clearly drivers. Other common aberrations in PI3 kinase are clearly drivers. They've been extensively characterized. There are a number of algorithms that have been demonstrated or developed to attempt to predict which are drivers and passengers, and I will tell you that they are not very effective. Indeed, we looked at the 9 current algorithms with 1,000 different aberrations from the Cancer Genome Atlas, and not one of them was called the same deleterious by all 9 algorithms. So we need better approaches, and this is a major program at the MD Anderson Cancer Center, and even more so in my own personal laboratory. Now, I want to go to a question that's a little further down the list, is what are the bioinformatics challenges? And is this an opportunity for career development? I'm going to be very excited in saying that even though we have great challenges right now with funding and positions in cancer in general, cancer research in general, the one area where there are major opportunities is around bioinformatics and being able to link biology, informatics, and patient care. This is critical. It is massive. We are generating data at a rate that we have never done before. And in reality, one of the major rate limiting challenges we have is in handling and analyzing that data. Now, I mentioned the concept that we can sequence a genome for $1,000. I actually think that is insufficient depth to be useful, but I can certainly sequence the actionable genome, that which I need to know about for $1,000. The estimate today, though, is it is going to cost us $100,000 to determine what in that genome is important to manage. That's partly why we've developed the personalized cancer therapy website, so that people can benefit from the major efforts and investment we're making in identifying drivers and determining what to do about them. But this is going to be the challenge for the future. Okay, so one of the questions that came up is one that I related to or alluded to. Do we need to obtain new biopsies on every patient? Can you get the information that you need from circulating DNA, and can you get that information from circulating tumor cells or imaging? At this point in time, the gold standard remains characterization of the patient tumor. I think that is critical, and we need to use that and continue with that as what we're doing. I think that the opportunity of moving to circulating DNA and circulating tumor cells is great, but that we will need to determine how and when that is effective and when it can replace biopsies. So for a while, biopsies are the standard of care. Okay. We have one or two more questions that we can answer. I'm going to leave it up to the organizers as to whether we can go a few minutes over in our broadcast time. One of those is to ask, are there new methods used to treat cancer? I think I've already alluded to this, and I want to come back. I believe that the future of cancer care is going to be integration of the methods that we're using today, but much better ways to integrate that information. So targeted therapies, going beyond what I talked about. I did not talk about transcriptomics, metabolomics, methylomics, all other opportunities that are arising, combined with targeted immunotherapy, some cases of stem cell therapy, and putting that into an armamentarium where we can pick the right patients to move forward. I want to finish with one final question, which is, what do I consider the minimum clonal rates of mutations in a primary tumor to worry about? And I wish I could answer this in a definitive manner. Right now, we use a cutoff of the aberration occurring in 5% of the tumor cells as the minimum amount that we can work with. When we look at the estrogen receptor abnormalities and ask whether a person will benefit from ER-targeted therapy, 1% of the tumor cells being ER positive is considered to signal an abnormality that is worthwhile treating. So right now, we have put a cutoff as 5%. I think as our technology improves, it may go on even better, and we can get down to 1%. But that is going to require clinical trials and a lot more work to answer the questions. So I think that I've now gone over all of the key questions that we have and responded in either directly or indirectly. And I would like to close this today and say thank you for your attention. And I hope this has been useful. These slides will be made available for you to look at your leisure at another time. Thank you.